it's my honor to speak on this wonderful forum. Um, so I'll start with a few introduction about our continent, and we'll give some example also how population migrates, and what kind of tools we are using, how DNA can be helpful to understand our deep population history. So with this, I would like to um, tell that we have archaeological evidences, and most of the time we try to validate or we try to corroborate the archaeological findings, and we try to fit our genetic data and genetic modeling into archaeological findings. So as we all know that modern humans evolved in Africa, and about 1 lakh years ago, or maybe 120,000 years ago, the modern humanity or modern humans, they started moving out of Africa, and South Asia was first geographical region where modern humans migrated and they settled down. So we use mitochondrial DNA, Y chromosomal DNA, and autosomal DNA. Now we have a lot of techniques and cutting edge scientific tools to sequence the Y chromosomal DNA, autosomal DNA, mitochondrial DNA, not from only modern samples, but we can also sequence the DNA isolated from archaeological remains. So the modern data, which was collected all over the country, that shows that about 100,000 years ago, modern humans started accumulating genetic diversity. So it proves and it confirms that modern humans started diverging among each other about 100 to 120,000 years ago. So mitochondrial DNA that comes from mother to offspring, and Y chromosomal DNA that comes from father to son, and autosomal DNA that shares equally from their parents. All kind of data shows that modern humans evolved in Africa and then started migrating to rest of the world about 100,000 years ago. So our history is very small. If we uh, measure uh, these dates on a global or on the scale of our evolution, so these dates are very small, and over the time of um migrations, a lot of mixings and lot of population replacement happened, and that led to the tremendous genetic diversity among modern humans. So this is the fact that modern humans have less diversity with chimpanzees. The chimpanzees, which are only found in Africa, so they have 3-4 times more diversity than all human being found across the world. So genetically, if you see the diversity of modern humans, so although we are very diverse, but if we compare our diversity to other species, so our diversity is very much limited. So as I said, modern humans evolved in Africa and they migrated to South Asia via southern coastal route. So we have three population sets in Andaman and Nicobar. They are named as Andamanese, Greater Andamanese, Jarvas, and Onges. So they are perhaps first settlers who have not mixed to other population groups in India, and they are isolated to Andaman Islands. So they were perhaps the people who migrated out of Africa about 70 to 80,000 years ago. So we use Onges and Andamanese data to model the ancient population group of South Asia. And like we all Indians, we have genetic components coming from these tribal populations. So they are our ancestors. So the genetic data confirms that Andamanese, they are the first migrant out of Africa, and then after that, several wave of migrations happen, and That migrations led to multi-layered population settlement, and that also led to different diversity matrices within India. So now we have about 4,000 well-defined population groups, which includes many tribes, many primitive tribes, and a handful of hunting and gathering societies also found. We have diversity in languages, social structures, dress and food habit, marriage practices, and all these barriers has given us tremendous amount of diversity. So it's very difficult to summarize the genetic history about our India in 30-40 minutes talk, but I will try to focus how this diversity has led to different population groups, and how languages and diversity goes along, and how genetic can be helpful to understand that diversity indices within South Asia. So in 2009, there was a big paper that came out from my lab only. Although I'm not the part of this study, but I am the witness of this big study. In this study, what they did, they have collected 25 population groups all across the country, and they have genotyped using autosomal markers, and they have concluded that majority of Indians, they are having two major ancestries. So they have given name ANI and ASI. So ANI, mostly North Indians, they are having ANI ancestry. ANI denotes ancestral North Indian ancestry, and the South Indians, they are having ancestral South Indian ancestry. So this ancestry was formed up to 5,000 years ago, and during 4,000 to 2,000 years ago, ANI and ASI mixing started taking place. And if you look at the current scenario, so all the Indians, they have ANI and ASI ancestry. So within this time period, 4,000 to 2,000 years ago, the ANI and ASI, they have mixed, and now our ancestry is like sandwiched. So we have some proportion of ANI and some proportion of ASI. So majority of North Indian and South Indians, they have this mixed ancestry. So there's no pure ancestry found anywhere in the country or anywhere in the world. So we all are mixed, and this mixing is not it's not a result of a single event. This mixing has taken place several times, and if you want to look at the deep rooted ancestry, so it's very difficult, because modern day South Asians, they are highly admixed, and they are the mixed population. So this ASI ancestry was formed like long back, and then ANI ancestry has some proportion of West Eurasian and Iranian ancestry. So that why so this ancestry is somehow different compared to ASI. So I started my journey. So I'm a trained molecular biologist, but I have very deep interest in archaeology. So first time I have collaborated with archaeologists and historians, and we try to collect data from archaeological remains. So the India has tremendous amount of samples collections all over the country, and despite of the wealth of enormous amount of samples, we are lacking trained manpower, and we are also lacking laboratory to analyze such kind of samples. So I attempted first time to establish this cutting edge research and this cutting edge facility within India, and we tried to retrieve DNA from samples from human remains who have lived about 4,500 to 5,000 years ago, and we tried to extract DNA from individuals. She has lived about 4,500 years ago. So we have been successful. We isolated DNA from this individual, and we have sequenced not full genome, but partial genome data we have got. So from this limited and marginal data, we have concluded that this individual is female, and this individual is having majority two kind of ancestry. So we have given this, we have compared the genetic data of this individual with all the published data sets, which includes ancient DNA data set from all across the world. And we have also we compared the data from Middle East, Central Asia, and from Swat Valley. And we concluded that this individual is having majority two ancestry. One ancestry is that we have given name Indus periphery, and the second ancestry is deeply rooted Iranian ancestry. So we also found that this individual is not having any Central Asian or steppe related ancestry. So first time it proves that the Harappan civilization was indigenous, and it was developed, it was formed indigenously by the local population groups. I'm not sure by that time there was a caste system or there were a distinguished group of populations, but that time the ancestry was common. So all across the Harappan area or IVC, the ancestry was, majority of the ancestry was common. And so the main ancestry was matching with the Andamanis related populations and the Iranians who were ancestors to the Iranian farmers and herders. So you know that the why and how Iranian ancestry appeared in the Rakhi Gadi sample. So we tried to date the ancestry. When did this ancestry arrive in in this individual? So we did a lot of modeling, and we have compared a lot of samples from Iran from different time periods. And we found that this ancestry, the Iranian ancestry, appeared in this individual about 11,000 years ago, before the advent of farming in Iran. At the same time, we did not find any Anatolian related ancestry. So it confirmed that Iranian ancestry appeared in Rakhi Gadi individual before the advent of agriculture. So from this data, first time we confirmed that agriculture system in IVC was indigenous, and it was developed indigenously, because we did not find any match with the Iranian herders or Iranian farmers. But this ancestry appeared long before the advent of farming. So this was a breakthrough discovery in terms of Harappan settlement. So we also found that 11 individuals from a site in Central Asia that was called Gonur and Sahare Sokta. So these sites were excavated in 2016-17, and 25% of the samples from these sites were outliers. So they were not matching with the remaining 75% of the samples. When we compared Rakhi Gadi data from these outliers, we found a match. We found that these outliers, they are more or less matching with the Rakhi Gadi individual. So it also shows that there were migration from IVC to Central Asia. So first time our paper has talked about these migrations. We also did a lot of analysis and modeling to understand whether it was bi-directional movement or it was a unidirectional movement. So then we confirmed that, so this migration was unidirectional. So a lot of population movement was already there about 4,500 years ago from Indus Valley region to the Central Asia. So so these results are making first case report about out of India migrations about 4,500 years ago. So there are like many line of evidences which I can't talk about that evidences here. But we have also compared the material culture that was found in Sare Sokta and Gunur, that shows that a lot of material resemblance was there from Balochistan and from this Mehergarh. So using material culture also, it confirms that this migration has taken place from IBC to this region. So this is the first case of like out of India migrations, and we did not find any steppe ancestry and Anatolian related ancestry in Lakhigadi individual. So it shows that that time the population structure was cosmopolitan and a lot of trades were already established, and a lot of trading was in practice from IBC to Central Asia. Coming back to late Harappan time period, we have samples from very well established and well excavated site, that is in Sanauli, in Bagpat district of Uttar Pradesh. So here, royal burial was excavated by a team of Archaeological Survey of India, and they have excavated human remains that was dating back to about 1700 to 1900 BC. From this individual, we try to extract DNA, although we have very marginal data, but that marginal data shows that there were no step integration by that time period. So from 2500 BC to 1800 BC, we did not find any Central Asian or step related ancestry integration in South Asian context. So we are also working on a lot of human remains that was found, that was excavated in Kashmir Valley. So you must be knowing there's a site in Kashmir very close to Srinagar. So this we call Burjahom Archaeological Complex. From this Burjahom Archaeological Complex, about 12 to 13 human remains were excavated from different time period, and that dates goes back to 4500 to 3rd 4th century AD. So we have human skeletal remains from 4700 years before present to 4th 5th century AD. We have been successful to retrieve DNA data from this Neolithic site, and data quality is super good, and we have very good quality of DNA evidences. And from this data also, we did not find any Central Asian ancestry until 1500 BC. So this work is not published yet, and this is unpublished data I am showing here. I have limitations because I can't show much data out of these samples, but this is making first case report that shows that until 13th 14th century BC, there were no steppe or Central Asian ancestry in South Asian context. We also found that Burjahom sample is matching with the IBC claim, and we also found that there was a continuity. So this settlement was not perished or not vanished. So the ancestry has continued until 2nd, 3rd century AD. And there is also a lot of match with IBC ancestry. So Indus Valley ancestry and the Neolithic Kashmir ancestry has quite resemblance. And in fact, we also found that the modern day Kashmiri, we have Kashmiri Brahmin data. That Kashmiri Brahmin data also has a lot of gene flow, and there's a continuity. So in Kashmir Valley, so there's a continuity since 4700 years before present until this time period. Although modern day Kashmiris, they have supplements of Central Asian or steppe ancestry, but that integration was not validates the claims which was put forward by eminent historians and Western scholars, that this ancestry has arrived earlier than 2000 years BC or 1500 BC. So I'm not going to make this topic controversial. I'm not going to talk about Vedic culture. Uh, I'm not going to talk about languages. But what we have data in hand that shows that until 13th, 14th century BC, there were no Central Asian ancestry appeared in South Asia. So here also we are also trying to reconstruct or revalidate the earlier finding in terms of South Asian well-established model. So this ANI and ASI mixing that was claimed that this formed about 4,000 to 2,000 years ago. So that we are trying to validate, and the modern data and the ancient data suggest that this ancestry was formed earlier, but that mixing date we have to calibrate again. So now coming to the gene flow that has taken place in India and the gene flow um, that that has taken place out of India. Although we do not have much data about gene flow out of India, but with limited data, I'm trying to make a good case study that we can have some hope to talk in this direction in future. So like India was a country where a lot of migrations has taken place. If you go back to 2000 years ago or 2500 years ago, we have a lot of records. We have a lot of West Eurasians and European ancestry that has been continuously integrated into our genome, and we have a lot of genetic records. We have a lot of historical records. But I will give you some example, and I'm giving here two example about some recent gene flow. This study was done in 2016, where it was found that with with Portuguese, lot of traders, lot of slaves were also appeared in India, and they have also contributed a lot of their ancestry. And if you go back to these populations, so say these populations ancestry is so diluted, you can't really reconstruct their original ancestry. So this is the case of Siddhis. So they appeared, they have come to India in 14th, 15th century AD, and now they have admixed with the local populations. So it's really hard to trace out their original ancestry. Similarly, we conducted one study on populations which are mostly found in northwest of India, and we compared the data with the Roma population of Europe, and we found that modern day Roma populations, which is mostly found in European countries, so they have their roots in northwest of India. So they migrated to European countries about second, third century AD. And if you look at their population number and their demographic structures, so they are up to 7-8 million populations which are presently found all across the Europe. Similarly, we have data from Y chromosomal markers that we call R1A1 line of ancestry. This kind of ancestry mostly we use to understand the male mediated migrations. So this R1A1 markers is mostly used to understand the male driven population movements. So until 2013 14, we did not have much data of this ancestry, but now we have collected data from more than 2,500 individuals, and we found that R1A1 ancestry is more diverse in South Asia. So far it was found, it was hypothesized that um, R1A1 ancestry um, was evolved in Europe, and since it is found in South Asia, so maybe the steppe or Aryans, they have brought this ancestry to India, and it was a concrete proof until 2014-15 to show that Aryans they came to India with this gene, and they have diversified R1A1 ancestry in South Asian context. But now we have data, we have huge number of data, and we have found that the diversity of sublineage of R1A1 and R1B gene, not gene, this R1B marker is high. The diversity is higher than the European population, and it shows that the R1A1, many branch of R1A1 was evolved in India, and perhaps it has migrated out of India. So this is not the case that Central Asians or the state populations they brought this ancestry or this marker to India, but it is the case that this ancestry, this marker evolved independently in India, and it got diversified over 5, 6, 7,000 years of time span. So all these evidences are coming day by day, and that giving different dimensions of genetic research, genetic findings, and it shows that whatever was taught, what was published, that need to be cross-checked, need to be revalidated. So coming to another site, that we have samples from Bali, Indonesia, and we have very good data sets from the individuals who have lived in Bali around third, fourth century BC, and we found that they have a lot of resemblance with the coastal population of Odisha and coastal population of Tamil Nadu. So it also shows that out of India migrations in third, fourth century BC from this region. So this is again unpublished data set. Coming back to one very interesting population group, which is mostly found in Kerala, and you must have known about Cochin Jews. So Cochin Jews are mostly found in Cochin, and there are Bene Israel Jews. They are mostly found in Mumbai, and there are a few Jew community. They are also found in Kolkata. So we try to understand how this ancestry mixed with the local population, and when this ancestry arrived into India. So we have collected data from like more than 500 individuals from Cochin, from Kolkata, from Mumbai, and when we compared data set from Middle East and European Jews data, we concluded that they arrived to this part of world in 7, 8, 9th century AD, and when they arrived, they started mixing to the local population. So in 40, 50 generations of time, you will be unable to trace out the original Jew ancestry, because that mixing is so significant and so strong that to trace out the original Jew ancestry will be very difficult. So I'm just putting this example because like a lot of migrations, the migration has never stopped. It was a routine practice since last 2,500 years ago. And using genetic data, one can reconstruct the original population structures, and when that population arrived to this region. So when we compare the Cochin Jews data with the published data set from Middle East, Europe, Central Asia, we found that about 30 to 40 generations before present, they arrived to India, and they have heavily admixed with the local populations. And now you can see here this the red color and this blue color shows that high Jew ancestry that is mostly found in European and Middle East country. But when you see in India, so the Jew ancestry is only 0 to 8%. So it got diluted significantly, and now genetically won't discriminate the Jew ancestry and the local ancestry. Similarly, Parsis. So we have one site in Gujarat. That site is known as Sanjan archaeological site. So in the Sanjan, we have found a lot of human skeletal remains from a burial site of Parsis, and basically it's a Zoroastrian cremation ground that we call Dokma. So in that it's like a tower of silence. In that well, they put their deaths. So from that well, we have excavated a lot of human remains, and when we analyzed the genetic ancestry of those individuals, we were shocked to see their ancestry. So the Parsi ancestry or the Zoroastrian ancestry is matching with the Neolithic Iranian ancestry. When you compare the modern day Zoroastrian or modern day Parsi ancestry, which are found in Iran, so their ancestry is more towards the Neolithic Iranians. So in terms of admixture events, when the Iran was not practicing Islam as a religion about in to 6th 7th century AD. So that time they escaped from Iran, and the Parsis they landed to Gujarat. And in Gujarat they maintained their ancestry. But in Iran, the Zoroastrians who are still in Iran, so they could not maintain their ancestry, and they had to admix with the population which came from Arab. So in terms of the original Zoroastrians ancestry, Indian Parsis, they are having the original Zoroastrian ancestry. So we are not only working to understand the genetic makeup of IVC and whether step is when this ancestry arrived and how population demography has been changed over the time, but we are also working on these small questions, and that shows that how India was a geographical land that time when lot of movement, lot of migrations took place, and it was a very safe land when populations they came, and without mixing, without forceful mixing or the consensual mixing, they have been here since that time, and they are maintaining their original gene pool. So these are the admixture plots. You can see like they still have a lot of Neolithic Iranian ancestry, and they are still maintaining that ancestry. So they feel proud that they are having that original Zoroastrian ancestry, and they arrived to India in 7th 8th century AD. They are also very highly endogamous. When we compared their endogamicity, we found that the Parsis are one of the highly endogamous group which are found in India, and they are now their census size is less than 55,000. We are also working in northeast of India, because northeast India is a land where we don't have much data, and there's a site in Shivsagar district of Assam where the Ahom dynasty was established, and they are very proudful warriors, and they have saved that territory from the Mughal invasion for a long time of time. So we have analyzed that data, and we found that they have very strong connection with Southeast Asia, mostly Thailand. So genetically, the Ahom they are carrying Southeast Asian ancestry. At the same time, they are also mixed with the local Assamese population. So that also like bridge the genetic the gaps between Southeast Asia and northeast of India. We found that like the majority of the Y chromosomal DNA that is coming from Southeast of Asia, and at the same time they have equal ancestry that is coming from the local population of Assam. So we are working on mitochondrial DNA and Y chromosomal DNA. We are trying to see the sex mediated ancestry. We found that they came, it was not a male, only male mediated ancestry, but it was equally like male and female, they both came, and they established their dynasty in Assam, and they mixed with the local population. We are also working to understand the genetic ancestry of, this is again unpublished work, of Nagaland and other northeast of region of India. And the data is super fascinating. We have samples which is dating back to 3,378 years before present, and we have three individual data in hand. So we are trying to understand the original inhabitants of Nagaland and how this population was changed over time, and what was the original or the ancient ancestry of northeast of India. So these are the cave sites. We have got samples, very good set of samples, and we have given the name PTL1, PTL2. We have very good data sets. All the data we have authenticated. They are ancient DNA in nature without any contamination. We found that the the ancient Nagaland populations, they have ancestry that is coming from majority from the three different regions. First is Tibet, and second is the Himalayan region, and third is coming from Yellow River civilization, which is presently found in China. We also compared data sets with the present data set of India, Nepal, and we found that they have like majority of the ancestry is coming from Tibet, Yellow River, and Himalayan region, and they have also like somehow ANI and ASI related ancestry. We also have a lot of ancient DNA data from Himalayan region, mostly from Spiti Valley. So we have samples from Lipa, Kannam, Sanglang, Tashiganj. So we are covering all the Spiti region, and we are trying to look at their demographic changes over the time. And we found that majority of the samples they have ancestry that fits into the ANI and SI model, and they are also continuing the populations. We did not find any significant changes over the time, and since the earliest settlement, these populations they are staying there and they are carrying forward their gene pool. So they have also majority of ancestries matching with the ANI-ASI, and they have also very good, uh, very good gene flow from Tibet inward and outward. So these are the admixture plots. They have also somehow very close ancestry with some of the population which is found in Nepal, and they have also a source population from Tibet, and remaining ancestry they are fitting into this ANI and ASI model. So I'm just giving one another example that I think that I thought to present this work also, because it has a lot of significance. This work we started in 2015-16, and then I thought to just give one glimpse of this kind of research also. So there's a lake in Himalaya. Mostly, this is found at 5,550-meter altitude in Kumayu region of Uttarakhand. In this lake, we found a huge amount of human skeletal remains, and there was a mystery. There was many speculations and hypotheses about the origin of these human skeletal remains, and then we started investigating about their genetic ancestry and what was the reason why this much of human remains are found at such a high climatic conditions, harsh climatic conditions, and at such a high altitude lake. We have analyzed 35 individual samples. So you can see the bones are scattered all over the lake and in the vicinity of the lake. We use ancient DNA extraction and whole genome sequencing approach to understand the source of ancestry and who are these people and what they were doing. So we did radiocarbon dating of 35 individuals, and we found that these individuals they have perished in 8th 9th century AD. So there was some harsh climatic conditions, and they were traveling to this terrain, and they got perished due to harsh climatic conditions. Surprisingly, we found two kind of ancestry. One was matching with the present day South Asians, and the second ancestry is matching with the Greek populations. So we were surprised, and still we have no answer, no clues. What and why what Greek were doing here at such a high climatic conditions. So with the advent of data, with the advent of add up of more and more data, we will have more and more questions about our complex population structure. So in this case of Rupkund, there's no any literary evidences, there's no historical record that why the Greek were traveling to such a harsh climatic conditions. So if they were here, they might have admixed with the local populations. So if Greek ancestry is, if you're finding Greek ancestry in some other population group, you please don't be surprised and don't misinterpret the finding that Greeks invaded India and they contributed their ancestry. So they are the recent samples, and we found that this ancestry was persistent since that time period. So we also found that this ancestry is very so we did not find much diversity in the Greek ancestry. So they are clustering very tightly all together. So they are from a same ancestral background, but the samples which is matching with the South Asian clan or Indian clan, they are highly diverse. So I just kept few lines just to I'm just trying to maximize the takeaway message from my talk. So I felt like difficulty in writing these lines because we can't summarize the genetic talk, genetic work in few lines, but still I'm just trying. So India is genetically not a single large population. Instead, it is best described as many smaller, isolated populations. Endogamy practice in India is very old and has taken place over the time. And the tribal and caste groups are not systematically different. Caste grew directly out of tribal-like organization. So we don't have much genetic differences between caste and tribe. And India is a melting pot which gave rise to several world population. We did not find any Aryan invasion from the data which what we have in our hand. And there's a very famous quote. I'm just keeping it here. So it is product of an unscientific, culturally biased form of thinking that saw race in terms of color. Scientifically speaking, there are no such things as Aryan or Dravidian races. The three primary races are Caucasian, the Mongoloid, and Negroid. Both Aryans and Dravidians are related branches of the Caucasian race. Biologically, both the North and South Indians are the same Caucasian race. Only when closer to the equator or the skin become darker, Caucasian race is not the white race. It can be of any color from pure white to almost pure black with every shadow of brown in between. This is not my statement. This I have just taken from internet, written by Thomas R. Trotman from University of Michigan, USA. So the few lines about the Dravidian and Vedic culture. The best Vedic chanting rituals are preserved in South India. Dravidians do not have to feel that Vedic culture is any more foreign to them than it is to the people of North India. They need not to feel that they are racially different than the people of the North. They need not feel that they are losing their culture by using Sanskrit, nor need they feel that they have to assist themselves against North India or Vedic culture to protect their real heritage. Meanwhile, the people of North India also need not take this North-South division as something fundamental. It is not a racial differences that make the skin of South India darker, but merely the effect of climate and gene. The Aryans and Dravidians are part of the same culture, and we need not speak of them as separate. Dividing them and placing them at odds with each other serves the interest of neither, but only serves to damage their common culture. Perhaps the saddest thing is that modern Indian politicians have also used this division to promote their own ambitions, though it is harmful to the unity of the country. So, the last but not least, India is the cradle of the human race, the birthplace of human speech, the mother of history, the grandmother of legal legend, and the great grandmother of tradition. Our most valuable and most instructive materials in the history of man are treasured up in India only. That was quoted by Mark Twain in 1835, who lived 1835 to 1910. So I am very much thankful to my collaborators who are working with me, and without the help of the collaborators, it's really very hard to generate this much of data. And I'm very much thankful to Sangam Talks to give this to provide this opportunity. Now I'm happy to take a few questions. Thank you so much.
So David Reich, the geneticist from Harvard, claims that he found genetic evidence of an influx of people from the Northwest in about 1700, 1800 BC, which was widely applauded as, oh, finally the proof of the Aryan invasion. To this, there are two answers. One is more my field, which has to do with Vedic literature, of which the chronology is such that this finding of an influx becomes totally irrelevant. Why? Because the Vedas are earlier. Sanskrit speakers were in India well before 2000 BC. So any influx that happened at the time is the same as the influx of the Scythians, the Greeks, the Huns, the Turks. They came in India, but they didn't change India's linguistic landscape. There is a second answer, though, and that is your field. And so is this true at all that there was an influx in like 1800 BC? Or would you say that, on the contrary, there is a genetic evidence of an emigration from India? What's your story?
Yeah. Genetically, if we talk about modern day Indians, most of the North Indians, they have steppe ancestry. And this ancestry is, in some population, this ancestry is high, in some population, this ancestry is low, but all the North Indians, mostly North Indians, they have this West Eurasian ancestry. So we are just trying to find out like when this ancestry arrived. So until 1800 BC, there are no such ancestry found in India. So whatever the data I have and we have analyzed in a very accurate way. So with a lot of confidence, I can share that until 1700 BC or 1600 BC, we did not find any such ancestry. So if genetic data corroborates the other field of evidences, we can definitely disprove this Aryan invasion theory. So far, genetically, I'm very much sure that until 16th, 17th century BC, we did not find any steppe ancestry. We have samples from different regions. I have samples from Kashmir, from Sanoli, from Rakhigadi. We are also collecting more data, but we did not find any steppe ancestry which is going back to 1500, 1600 BC samples. although we have lot of samples which is matching um, perfectly, that is coming from Central Asia. And as I said, 25% of samples which was found in Turkmenistan and east of Iran, and those samples are going back to mature Harappan time period, and the date is around 2300 to 2400 years before present. So in fact, we are finding Harappan ancestry in those places at that time period. But so far, we did not find any samples in Indian context which is showing steppe or Central Asian ancestry in that time period.
Mhm. In that case, the Bactria margiana culture, what is its genetic relation with the Harappan area? Either way, you see, it could be an emigration also, but so is there any sign of this migration?
Migration from which, out of India?
Yes, let's say migration from what is now Punjab to what is now northern Afghanistan to Turkmenistan.
Yeah. So actually, we try to understand whether this migration was bidirectional or as a unidirectional. So we confirmed that this migration was unidirectional, and we also found a lot of material culture in Turkmenistan that was completely matching with the IVC material culture. So using material culture data, and we have also modeled our genetic data, we confirmed that this migration was unidirectional. And the best part is that there was a cultural assimilation also, because if someone is moving out of India and that burial was found exactly in such a way that the original inhabitant of those areas were buried. So it was not like a accidental finding. So it was a burial. It was a symmetry. So in such a such a perfect manner where their original dates were buried, they have also buried the people who have migrated from IVC. And they have also buried their cultural artifacts, their potteries with the dead bodies.
Right.
Yeah.
Okay, a final question. Maybe more people, but certainly Premendra Priyadarshi has argued that there is a lot of genetic evidence for migration from India, though he doesn't cite any human evidence, but he cites the evidence of mice who follow the human beings and of cattle who are taken along by cattle herders, which is what everybody says the Aryans were, so that you find a certain percentage of Zebu genetics even in Syria and even in Ukraine.
Yes.
So can you further enlighten us on this?
Yes, I have collected cow breeds all across the country. So we have data from cow breeds for more than 12 breeds, including this the dwarf cow breeds of Kerala to North Jebu breed. So we have found that thus the genetic signals, which is like you mentioned in Syria and some of the taurine breeds of Spain and Italy, we found the Jebu signal in that genetic data. So the cattle data, it confirms that there was not exchange, but there was a migration, there was a movement of agro-pastoralist community out of India that time period. But we have genetic evidence from cattle, but we have also evidences from human remains also. So like and we have evidences, archaeological evidences also. We have evidences from material culture. So all these evidences are making good sense of out of India migration. But at the same time, if you compare ancient DNA data from Indus Valley or Indus periphery, we did not find much genetic contribution from IBC clade to the populations who were there in their time period, like Neolithic Iran or Mesolithic Iranian context. So there was not a human genetic contribution much at that time period. So here, at this point, we are lacking. We do not have much mixing signals. So what I am mentioning here, that there was a migrations. We found that Rakhi Gadi or IBC signal in Turkmenistan, but they were first generation migration. They have not mixed with with their populations. So until we are finding a genetic mixed data, we cannot confirm, we cannot prove that there was a mixing, there was a like settlement. So we are working in this direction. Hopefully, with more data, we'll have more evidences.
Okay, thanks a lot. Huh?
So the first question is that, sir, suppose there are similarity between population of A and B. So from that similarity, how it can be computed the direction of migration of the culture? There can be a possibility that the ancestor from A has moved from point A to B. So there will be again similarity after 100 years in if we see the genetic data of A and B, and again the vice versa is also possible. So how you compute, or how that is being computed?
Yeah. So actually, we know the ancestry, because if we are comparing the ancestry of Sare Sokta and Gonur in Turkmenistan, so we know the ancestry of hunter and gatherers populations who have lived 10,000 years ago. We know the ancestry of 8,000 years ago populations. We know the ancestry who have lived 6,000 years ago in that area. And we know the ancestry who have lived 4,000 years ago. Okay. So when we compare data from 10,000 to 4,000, and when this mixing happened 5,000 years ago, we are finding, like, for example, 5,000 years ago, we are finding that genetic similarity. But when we know the genetic makeup from 10,000 to the modern time period, okay, so if this migrations has taken place, if they would have mixed there, they could have contributed their ancestry, okay, but they have not mixed, so they have not contributed, okay. But if that ancestry is coming to this region, so definitely that ancestry we could have found in modern day South Asians. So we did not find that ancestry in modern day South Asians. So that confirms that that movement was not vice versa. So for example, if that ancestry 5,000 years ago, if those populations, they have Anatolian ancestry, okay, but we are not finding Anatolian ancestry in South Asians that is going back to 5,000 years ago. So that disproves that the migration was not vice versa. Another proof that we have, if somebody is coming from that side to this side, so they will definitely bring their material culture. So we did not find any material culture in that time period, but we are finding material culture from IBC side to those side. So that confirms that this migration was unidirectional. And despite of this, we have like a lot of models. So we do a lot of permutations, like simulation studies and permutation studies, just to see all the possibility, just to disprove, like we can't leave any possibility, like we have to see like all kind of possibilities. But with all possibility, we did not find any hypothesis that is fitting inward movement.
One of your paper that was in Science, so the formation of human population in South and Central Asia. So there a claim has been made that this Yamanian steppe pastorals have came 1,000 years up to 1,000 years BC to contribute ancestral North Indian.
Yeah.
And another paper in BioR 14, there was also in the abstract, there was a claim that the population means from the genetic responsible for the spreading of Indo-European language across much of Eurasia. So are this claim are not contradictory? So what you are saying, because this is in 2019 and 18.
Yeah, this claim is contradictory, and the language and gene doesn't go along. So if you have genetic signatures or genetic findings, genetic proof, then you can correlate with the linguistic proof. So as I said, everybody has this step ancestry in modern day Indian population. So this is the question, like when this ancestry arrived. So I used to believe that this ancestry must have arrived in 2000 BC time period. But as soon as we have data, we are just narrowing down. So I don't have any problem with step ancestry in 2000 years BC. In fact, couple of years back, I used to believe that this ancestry must have arrived that time period. But as soon as we are getting more and more data, we are just finding that this ancestry is not so early, but it's very late arrival of step ancestry in South Asian context.
It was a great talk, Dr. Rai. Thank you so much.
Thank you.
I have a couple of questions, but I'll keep it short. But if possible, I would like to email you further on. There are some more things I would like to know. So to get to my question, before that, I was very happy to see the data that you showed about Bali and Indian travelers to Bali. So taking off on that, actually, from all the research that we have been doing, we see that most of the texts from Southeast Asia, like all the regions across the Indian Ocean realm, like Java, Cambodia, Sri Lanka, Maldives, a lot of places, they actually have a lot of documentation about people from Kalinga going there and settling colonies, even Burma, in Burma. Like a lot of these places, rather we don't have in present day Odisha we don't recollect those, those connections, but they have in their ancient texts. I'm sorry. So reading upon some very old books written by, like from the translations of those ancient texts, I found that. So do you have any further plans of taking a bigger data scale from the entire erstwhile Kalinga coast? Kalinga coast, as you know, was from Kalinga region, was from the Tamralipti area to all the way till the Krishna Delta. So right now they are all different, named as different states and all, but the entire Kalinga region, they had a regular exchange of people and culture through to all the South Indian Ocean regions. So do you have any plans of taking more data points throughout the Kalinga coast, along with all these places of Indian Ocean realm, like even to Maldives, to farther reaches of Oceania and Burma, and all these places? So do you have anything like that in the plans? And
I think I would be happy to collaborate with you on this topic, because we are trying to collect a lot of informations online, and we are also exploring libraries. But we are finding a lot of evidences that, as you mentioned, that from Kalinga regions, a lot of maritime trade was recorded in the literature of Southeast Asian countries. But we are having a lot of informations, but we are just struggling to compile that information, which can be useful, which we can use in our publications or our purpose. So I think for this, I would love to collaborate with you, and if you have time, we can set up a meeting, a discussion in coming future.
That would be wonderful, sir. I would email you, and I have just one more question. Mm-hmm. That is related to your. It was again something very significant I saw in your presentation today about Himalayan regions, the genetic data you showed. So here I need to share something first, and then I'll ask my question. Mm-hmm. So many people don't realize that during the Vajrayana period of Buddhism, like right now, if you see in the Himalayan kingdoms of Nepal, Bhutan, Tibet, all the Himalayan kingdom, the Buddhism that is followed is more of the Vajrayana Buddhism. Mm-hmm. And many Siddhas and Mahasiddhas from Odisha, actually Odisha region, the Udyana region, they went to the Himalayan kingdom. Like they carried this Vajrayana. In fact, Padmasambhava was the adopted son of Indrabhuti, who was the ruler of Sambhala. His text starts with a invocation to Jagannath himself. So my question here is, have you ever thought of this very niche area of doing genetic studies of this region with the Himalayan kingdoms? Because there was outflow of a lot of, and inflow too, but outflow of a lot of people during the Vajrayana period.
Yeah, so very good point. You have mentioned a very good point. There are samples which we are analyzing from this time period. So what's the date of this Vajrayana Buddhism that appeared in Himalaya? I think after 6th, 7th century AD.
I think so, yeah. Because
So
Indrabhuti, yeah.
The late samples, which are dating back to 6th, 7th, 8th century AD, we are finding this ASI ancestry. So in Himalaya, ASI ancestry at that time period must have brought by the populations from Odisha, maybe during that time Vajrayana Buddhism expansion. So that we are just trying to explore how this ASI ancestry could have arrived in that time period. But the samples which are going back to early time period, like say 2nd century AD or 1st century AD, we are not finding ASI ancestry. So that ASI ancestry in 7th, 8th century might have brought during this Buddhism expansion. So that we have to validate. We can't really prove that this ancestry might have arrived due to this Buddhism expansion, but we can make a very strong case, and we can discuss about this religious expansion.
Do we have any AASI DNA samples? And also, when do we first get Indus Klein ancestry in the deep south? Estimated admixture date, what could that be? Thank you.
Unfortunately, we do not have ancient ancestral South Indian ancestry, but we have data. The data are coming from South India, so that data is coming from 5th, 6th century BC samples. So from this, we are going to figure out the ancient ancestral South Indian ancestry, and that will publish this finding soon. And also I'll talk maybe sometime in some of my next lecture. But so far we don't have ancient ancestral South Indian ancestry, because all the South Indian populations, they are mixed. So although we have very few populations like Palayan, Mudaliyar, and Palliyar, these are the populations. They have more ASI ancestry, and they could be a good proxy for ancient ancestral South Indian ancestry, but we are not very much sure that without ASI ancestry, that we can't answer the exact date of that ancestry. So that I think to answer these questions, we have to wait. We need more data.
Sir, my question was that you was saying that some more publication will come in future, and that happened means 2 years early, one of your video, but the findings are not been published yet. So when they will be get published? So
yeah, with the publications like I mentioned, like R1A1 paper that is in pipeline, and we have megalithic data from South India. We have samples from Tamil Nadu, Karnataka, Telangana, Andhra Pradesh. So we have data sets from this 4 states, and the samples are very unique, and they are very quite old. They are from all megalithic context. So this paper is also almost ready, and then we are just interpreting the data. Hopefully in this year, both paper will arrive.
So, sir, this one of the professor of Harvard, Michel Visser. Sorry, am I pronouncing the name wrong? Maybe. So in one of your paper that Cell, so he have commented that of course there will be no steppe ancestry or R1A1, particularly the R1A1 gene, because that mixing happened after the decline of Indus Valley civilization, and the remaining is from the Indus Valley civilization. And again, some of the group has argued that this, as there is no presence of ARVN gene, although it was you said that it was a woman skeleton, so the problem is there. So, although the ARVN gene is not present, so they have concluded that the Aryan migration has happened after the Indus Valley decline. So, what will be your comment on that?
Yeah, that's why now we are coming up with more data. What we are finding that R1A1, the many branches of R1A1, that is deeply rooted to South Asia. And when we are analyzing more markers like G93, G95, and M270, there are many markers. When we are sequencing it and genotyping it, the downstream markers, we are finding more and more diversity in the South Asian populations. So if the diversity is very high, you can't say that this migrations would have taken place after the collapse of Indus Valley, because if diversity is very high, so it means that that haplogroup is old. So if that haplogroup is old, then how you can justify that this migration happened after the collapse of IVC? So like for example, if the downstream marker of R1A1, there's like R1A1, A2, A like something like this. If somebody is arguing that this haplogroup might have come through Central Asia around 4,000 years ago, but when we are estimating the age of this haplogroup, if that haplogroup is 6,000 year old, 6,000 year old before present, if this haplogroup is diversified, then how we can say that 4,000 years ago somebody brought this ancestry to South Asia? So this is the problem with the this Y chromosomal based haplogroup population studies. So until we are not very sure about the downstream line, because like R1A1, R1A, 1A, 2B, something like this. If this is the terminal of any tree, if we are not finding any downstream markers, downstream branch of that haplogroup, so until we are not finding, we can't be very sure. If we are finding downstream diversity very high in South Asia, and that diversity is very less in Europe, that we are speculating that during that time period, these populations or this haplogroup might have come. But we are not finding that diversity in European populations. But that downstream diversity we are finding more in South Asia. So that will be the opposite case.